on the attendance paper. For those who are online, I hope you can hear me. If there's some technical issues, please inform me also. So before we start about the today's topic, let's discuss some points from the colloquium, as well as about the following organization of the course. So about the colloquium results, in general, the results were quite good. Of course, there were some variations. Some did the colloquium a bit better than others. Some technical details. So in your e-studies, you should see the colloquium grades. Unfortunately, there's a problem with the IT department and they cannot add an additional column for the practical part. But the practical part results are with me. If you're interested in your practical part result, at the end of the class, they can really go through the table and tell what was the result. If you're interested in more detailed analysis, we can discuss that during consultation times if needed. But in general, practical parts are quite good. What else? So starting from today, we will have hybrid classes. So I already sent emails to the group leaders, but the idea is you have the option to come here on site to check the classes as it was in the first half of the semester. But those who already have the 50% attendance, they are also allowed to connect online. But the thing is, you can still come to all classes on site if you like. And you need to choose. If you're not coming on site, then you definitely have to be online. If you're not online, you need to come on site. So we are still checking the attendance. It's very important for those who still do not have this 50% attendance. I hope most of you will get this now. But if there's some issues, we will see what we can do. Okay, what else? I checked your protocols. Most of them were fine. There were some protocols that are missing, but I'll write individual emails or ask individual students about some of those issues. So today's topic, we will talk about development of the face, development of the skin, and also a little bit more about adrenal gland, the metanephros development. So a continuation of a previous topic. We will check slides, both from the slide pictures that are in your e-studies together with the microscope. So I will share the screen so everyone that's on Zoom could see this information, as well those that are here. If there's enough space, it's recommended that you can move maybe closer to the front rows that you can see the screen, because I will share the screen quite a lot. So everyone on Zoom could see what I'm showing. Also, you that are here on site could also see what I'm showing to you. So let's start. First slide that we will check will be about the oral cavity, development of the tongue and sublingual gland, week 6-7. So tongue development, week 6-7. That will be the first slide. So yes, there's a question on Zoom. If there's questions, better write in the chat because it's quite hard to hear you. So if there's questions, it's highly recommended that you write in the chat so it's easier for me to follow and hear and see what you're asking. Okay, I will start sharing the first slide. The development of the tongue, 6-7 weeks. This is the first slide that we will check and we will go through it in detail. So first of all, what is the main issue? Size of the slide. So the slide is quite big. I will reshare the screen so I could also show you the microscope. In the microscope, you basically see only a small area like this, for example. So you don't have a very large vision field. If I switch to the microscope, this is what you can see on the lowest magnification. So the slides are again quite big. But let's return back. So I will first discuss this whole picture and then we will move to the microscope so you can understand the differences. First things first, you should understand where the tongue is. So as you can see, you have the tongue. And the first thing that we will label in this tongue is here, number 1. That's the dorsal surface of the tongue. Dorsal surface of the tongue. That's number 1, dorsal surface of the tongue. So slide name, tongue development, weeks 6-7, and first structure, dorsal surface of the tongue. What you need to notice here, no tongue papilla. No tongue papilla. No papilla have yet developed. No tongue papilla. So no tongue papilla, the surface is smooth. Those who have come here, please come take the attendance paper. So write your names down on this paper, please. So no tongue papilla, that's the important aspect here. So smooth surface. Second, do not forget information that we already discussed in the previous colloquium. So as you know, tongue contains developing skeletal muscle tissue. So you might see reddish fibers in cross sections, longitudinal sections. So all of these reddish fibers inside, that's skeletal muscle tissue development. So developing skeletal muscle tissue is also here. So do not forget this. Then what else is in the slide? This area here. So if you check this area with number 3, we call this developing sublingual gland. So developing sublingual gland. So number 2 was developing skeletal muscle tissue. And number 3, developing sublingual gland. What you should notice here, so this sublingual gland, I will delete this message. So sublingual gland is made only from ducts. So only duct system has developed in week 6, 7, only ducts. So developing sublingual gland only made from ducts. Okay, developing sublingual gland, you only see the duct system, no secretory portions yet, so I hope you can see this on the screen. What else? We see also partially some structures from the oral cavity. For example, we see oral cavity epithelium. So 4, that's oral cavity epithelium. Oral cavity epithelium. So oral cavity epithelium, we can also see it. Sometimes it's thicker, sometimes it's thinner. By its structure, it's stratified squamous non-keratinized epithelium. But oral cavity epithelium is also seen in these slides. And below the epithelium, there's mesenchyme. So number 5, that's mesenchyme. Mesenchymal tissue. In your slide description, it's written mesenchymal papilla, but you can just write mesenchyme. Mesenchyme. Number 5, mesenchyme. In the oral cavity region, in the facial region, mesenchyme is also called ectomesenchyme. This term also appears in some textbooks, ectomesenchyme. It's mesenchyme of the head region. So it develops from the neural crest. So from neural crest origin. So it's a bit unique in this way. So ectomesenchyme, it can be used in this slide as a synonym for mesenchyme in the developing facial region. What else? You might see some other structures like this one, number 6. The early tooth development. So early tooth development. Early tooth development. So early tooth development is also visible in slides. It's in bud stage. In your slide description, it's written cap stage, but in most slides, it's actually the bud stage. Bud stage, early tooth development. If you write early tooth development, it's also completely valid answer. So you don't need to differentiate the stage, so you see this tooth development. Then, what else? As you understand, this whole area shows the lower jaw development. And what is inside this area, we will talk now in detail. You can see there's a small plate of cartilage. So there's degenerating cartilage tissue. And with number 7, when we call this apoptotic Meckel's cartilage. Apoptotic Meckel's cartilage. So from theory, what you should know, so as the facial region develops, so it also has some cartilage tissue, but this specific cartilage degenerates completely, so it disappears. As you know, bones of the face and the jaws typically develop by intramembranous ossification, so directly from mesenchyme. So normally there's no cartilage left. And this cartilage is only a temporary structure. It disappears quite rapidly. So apoptotic Meckel's cartilage. Next to this cartilage, you still might see reddish colored bone trabecules. Number 8 would be intramembranous ossification. Intramembranous ossification. Intramembranous ossification or bone trabecules. So intramembranous ossification appears around the Meckel's cartilage. So you might see bone trabecules here. So bone trabecules can appear in slides. What else you should notice? As I mentioned, there's also the lower part of the face. So you need to describe also some other parts. For example, you see this whole structure, we could describe this as the lower lip. So this is the developing lower lip. You see the lower part of the face. You see also the lip developing here. So developing lower lip. And because it's the lip, remember from histology, lip always has two sides. One side is from the oral cavity side, and the other side shows skin development. So here you can see developing epidermis, that's number 10. We will talk in detail how this happens today. So 10, developing epidermis, developing epidermis. And below the epidermis, this mesenchyme is developing dermis. So developing dermis. Hello, take seat. So developing dermis. Developing dermis. So number 10 is this darkened epithelium on the surface that's developing epidermis, and the mesenchyme below this developing connective tissue that's developing dermis. You might also spot a very dark structure coming out from the epidermis. That's developing hair follicle. So number 12, developing hair follicle. Developing hair follicle. So developing hair follicle. So hair follicles, you see these dark dots or specks of tissue that go into the mesenchyme, so developing hair follicles. Because it's the lip, you will see skeletal muscle tissue development inside the lip. So don't forget histology. Skeletal muscle tissue develops here on the inside, but you can use another name, musculus orbicularis oris development. Like in histology, so the lip has some similarities. So here also musculus orbicularis oris. If you write here skeletal muscle tissue development, that is also a fully correct answer. That also works. But musculus orbicularis oris develops here. So developing musculus orbicularis oris. In general, that would be the main information in this slide. So that's the main information you should know. That's the general idea. Is this slide understandable to you? Yes? So about mesenchyme and ectomesenchyme as well, the answers, yes, both of them are valid answers when talking about this specific slide. So about when we talk about the tongue development, yes, you can use ectomesenchyme to describe mesenchyme. Yes? You can say bone trabecules, that also would be a valid option, then add developing bone trabecules. If you remember intermembranous ossification, this term can also appear a little bit in the test. So don't forget this term. That's why we use this, yes? So colleagues, yes, please. Develop nervous system. Epidermis, yeah, epidermis is the developing surface here. What I said though? 11, developing dermis. 11 is dermis, 10 is epidermis. Sorry if I misspoke. So number 10 is epidermis, and then we have the dermis 11. Okay, I will show the microscope slide. So I will delete these drawings. If you want, you can take a picture or you can take a screenshot. Those are all for those that are online. If you don't, I will delete this. Okay, I will delete this. So next, I will show you the microscope. So in the microscope, it can look a bit different. I will use the green arrow to point at structures. So first, I will start at the tip of the tongue. So the tip of the tongue is here. The slide is a bit on a different angle, but still we can understand it. So we see the tip of the tongue, and we go along the dorsal surface of the tongue. So you see it has a smooth surface, no tongue papilla yet. So this is the dorsal surface of the tongue. Inside here, you even see developing skeletal muscle tissue. You see fibers. Here in this slide, these fibers are more purplish or bluish, but you don't need to check the stain. You need to check the types of structures. The skeletal muscle tissue is also developing in this tongue. You see muscle fibers here. Then if we go below the tongue, you see there's a developing duct system. This area shows the developing sublingual gland. So sublingual gland is made only from the ducts here in this slide. So developing sublingual gland. You can see it in this slide quite well. If we go a bit further, so we also see this Meckel's cartilage. So apoptotic Meckel's cartilage, you see chondrocytes here. The generating or hypertrophic chondrocytes are here. So you see chondrocytes, so it's cartilage tissue. So that's the apoptotic Meckel's cartilage. And here around in this slide, you also see bone trabecules. Here in this slide, bone trabecules look dark blue or purple. As I mentioned, don't check the color, check the position and the texture of these structures. So it's easier for you to understand them. What else? So Meckel's cartilage was here. Intermembranous ossification is around. Let's move closer to the oral cavity surface. So you see a bit of oral cavity epithelium. Here, for example, it's a bit fragmented, but it's still oral cavity epithelium. Below, there is mesenchymal tissue. If you write ectomesenchyme, that also works, but if it's mesenchyme, that also is a valid option. Mesenchyme. And here below, early tooth development. So this is early cap stage or late bud stage, but you see early tooth development here. Let's move on. You also see this lower lip. And for the lower lip, remember, one side shows the oral cavity surface, while the other side shows skin development. Developing epidermis is here on the surface, developing dermis below. So developing epidermis is here, developing dermis here. Unfortunately, we don't see very nice hair follicle development, but there's some developing hair buds, so these hair follicle primordia are still developing here, so very small primordia here. Yes? Oral cavity epithelium. This is oral cavity epithelium. Yes. And here in the middle, skeletal striated muscle tissue. Skeletal striated muscle tissue, or you can write musculus orbicularis oris. Yes? You mean these ones? I will zoom in. This looks like muscle tissue. Yeah, it's muscle tissue plus some blood vessels. So blood vessels with muscle tissue. It's still skeletal muscle tissue. I'm showing also these darkened slides that maybe are a bit different than these ones, so you understand the variations a little bit. In general, that would be it about this slide. So not the most complex slide, but again, be careful with artifacts. You can see sometimes the epithelium can be damaged, but it's still all oral cavity epithelium. Any questions about this slide, or we can move on? No questions in the auditorium, maybe some questions in Zoom. I see no comments yet, so let's move. Next slide will be tongue development, weeks 10 11. So tongue development, weeks 10, 11. So I will find a good slide variation for this. Let's check this plate here. So tongue developed 11, 12 also. Yeah, it can be used 10, 11, 12. There's different variations, but 11, 12 weeks. This one. I will put it on the microscope, but again, I will start with the e-studies. Sorry. Okay. So before we start checking the microscope, I will show you the e-studies picture. So in the e-studies, the slide looks like this. What are the differences? First of all, the size. So tongue in 11, 12 weeks is much, much larger, twice as large. So in the microscope, actually on the lowest magnification, the tongue will look like this. So it's a much bigger slide. That's first thing that can also help you in distinguishing it. But let's talk about other details. First, dorsal surface of the tongue, the same dorsal surface of the tongue. But here, the dorsal surface has developing tongue papillae. So primordia of tongue papillae. Here you can see the surface is uneven. These tongue papillae are developing. So developing tongue papillae. So again, number one, dorsal surface of the tongue. And in brackets, I will write developing tongue papillae. So dorsal surface is uneven. So the primordial tongue papillae are appearing in the 11th, 12th week. Then what else? Some structures will reappear. For example, skeletal muscle tissue. So developing skeletal muscle tissue, you can even see fibers. You can see quite a lot of skeletal muscle tissue fibers inside. Developing skeletal muscle tissue. So developing skeletal striated muscle tissue. That appears again. You might have a question about what is the tissue between the muscle tissue. So you see light staining tissue here. It's embryonic connective tissue or mesenchyme. Mesenchyme. Mesenchyme is also in the previous slide. We showed that in the mucosal region, but basically in every location where you have connective tissue, that you can call it mesenchyme. So mesenchyme is also not only next to the surface of the mucosa, it's also in between muscle fibers. So remember this, mesenchyme. What else? Here again, we can spot an organ. This is again developing salivary gland, or developing sublingual gland. So number 4, developing sublingual gland. But what you need to be careful with, so when we talk about this developing sublingual gland, we can distinguish 2 different parts. You can distinguish the ducts, so developing ducts, that's number 5. So developing ducts of the sublingual gland. So these are developing ducts. And 6, developing secretory portions. Developing secretory portions. Developing secretory portions, number 6. Maybe it's a bit difficult to see because the computer is in front, but there's small secretory portions. So small secretory portions appearing. So in 11, 12 weeks, the gland is much more developed. It's much more developed gland. Then in your e-studies written main excretory duct, you can just write developing ducts of the sublingual gland. That also works as a valid answer. It's not always necessary to distinguish which type of duct. It's not always possible. If you just say developing ducts of the sublingual gland, it's also a fully valid answer. Then what else? Only in this slide, you might spot small areas here. Number 7. These are nerve fiber bundles. Nerve fiber bundles. Nerve fiber bundles. If we would zoom in, you could actually see nerve fibers here. Nerve fiber bundles. So number 7, you see small nerve fiber bundles next to the sublingual gland in this region. It's not always in every slide, but in many slide variations, this can appear. Nerve fiber bundles. And then a very important detail. So in tongue that is 11, 12 weeks old, you might spot small glandular tissue. You see very small glands that are at the base of the tongue, so closer to the root or the base of the tongue. And these glands, with number 8, we call them developing minor salivary glands or developing lingual glands. So I will write developing lingual glands for short. These are minor salivary glands of the tongue. So developing minor salivary glands or developing lingual glands. Both are valid answers. Both are valid answers. In your e-study, it's written minor salivary glands, but you can also use a shorter name, lingual glands, developing lingual glands. If you see glands like these inside the tongue, it also tells you this is tongue 11, 12 weeks. These glands are not in the 6, 7 weeks old slides of tongue. So only here. And in general, that's it. That's the main information. Of course, blood vessels could be asked. No, I will zoom in some areas. You might spot arteries, veins, so blood vessels can appear in this slide. Some arteries, like here, there's a small artery. There's a big vein near that sublingual gland. So blood vessels also can appear in some slide variations. But in general, that's the main idea. Is this slide understood? Yes? What's the difference? Location and type of gland. So if you know from histology, we have 3 main large salivary glands. They are big clumps of glandular tissue. Minor salivary glands are scattered throughout oral mucosa, including tongue surface. And these smaller glands, they are much, much smaller. They are in this specific position of the tongue. So minor salivary glands are separate from the sublingual gland. These are completely separate glands. That's the idea. I see a question. So can I show the artery again? Yes, I can show. If I zoom in here, you can spot an artery. This is an artery. So that's the first question. So that's an artery. Now I will clear this. Then, what the next question? Are the structures on the left of the secretory portion nerve fiber bundles of the skeletal structures on the left? On the left. So if you mean these structures here at the bottom, yes, that's skeletal muscle tissue. So there is muscle tissue also below the tongue in the developing facial region and the jaws. That's also skeletal muscle tissue here below. Skeletal muscle tissue. Okay. Do we need to know the type of epithelium on tongue surface? So the epithelium on the tongue surface, stratified squamous epithelium. It's non-keratinized. In any location, even on this lower part of the tongue, this bottom surface, it's still stratified squamous non-keratinized, but it's not necessary to write the full name of the epithelium. I typically do not ask the full name of the epithelium. I ask you in general the structures, so the dorsal surface, the tongue papilla, etc. But it's stratified squamous non-keratinized epithelium. Okay, if this slide is understandable, let's move to the microscope so we have enough time for other slides as well. So I will clear the drawings and I will show you the microscope again. So a warning, this slide is very large. So it's a very big slide. And because of this, there might be some issues understanding some details. So I will put the chat away so it doesn't disturb anything. So a very large slide. So, but first things first, even in this magnification, you see dorsal surface of the tongue is uneven, so there's developing tongue papilla. So you see this very wavy surface, so developing tongue papilla. That's the first thing. Second thing, if I move through the whole slide, so there's an artifact, don't worry about it, but there's a very big area that the slide covers. This is the lowest magnification, so very large slide. You see inside the tongue developing skeletal muscle tissue. In the colloquium, I typically won't focus on the muscle tissue structure, which we already had them in the first colloquium, but still do not forget that muscle tissue is developing here. So developing muscle tissue is here. And if we go on the lower portions, there is an artifact, unfortunately. Unfortunately, the break in the glass has happened in not in a nice location. I will move to another fragment. There's also another tongue here. And let's check where the sublingual gland is. Okay, here, this is a better area. So developing sublingual gland, don't worry, that's a dust particle, it's an artifact. So if you zoom in, so there's developing ducts. So there's a larger duct that's developing, so developing ducts of the sublingual gland and developing secretory portions. You can actually see developing mucous tubules here. So you see very light staining cells, mucous tubules are appearing. So here your previous histology knowledge can be helpful. So sublingual gland is mainly a mucous gland. You will see mucous tubules. Mucous tubules. If you have not written attendance, please write. Okay, mucous tubules. Also notice that there's a lot of embryonic connective tissue, mesenchyme. Mesenchyme is also in between the developing secretory portions. Yes? It depends on the slide. Here you see a very big duct here. So the question was about the ducts. Do we see only a single large duct? Do we see other ducts? It depends. Here we see a big duct here, but actually if you go through the slide, you might spot some smaller ducts. You see there's even small elongated duct sections here in the slide. So there's different kinds of ducts, but typically if I point at structures here, I'll point at obvious locations, like here, that's a developing duct of a sublingual gland, and these are developing secretory portions. So there can also be very small ducts inside, but it depends on the slide and section. The epithelium. Ducts, first of all, have thickened epithelium. They're quite dark, and fully developed secretory portions are light staining cells. The coloration helps. So that's the difference. Okay, let's move on. There's a question in the chat. I will answer it afterwards. So let's finish the slide. Then next to here, we can spot nerve fiber bundles. Here, you can actually see how these nerve fiber bundles look in high magnification. So nerve fiber bundles next to the sublingual gland. So nerve fiber bundles can appear in slides. Nerve fiber bundles. Then let's move on. Let's go to the root of the tongue. So let's move to the root of the tongue. No, that's approximately here. And you start to spot small glandular tissue fragments. So these small glands are developing lingual glands or developing minor salivary glands. Both names can be used in the colloquium as an answer, but these are minor salivary glands or developing lingual glands. So you can use both names. If it's easier to remember developing minor salivary glands, it's a completely valid answer. And basically, that's it. That's the general idea about this slide. I see there's some questions in the chat. I will answer them, and then we will move to another slide. So I see there's a question. Are the big ducts interlobular ducts at this stage? No, these big ducts are not interlobular ducts. These are typically the larger main ducts. These could also be the main excretory ducts. Interlobular ducts technically could develop also here, but you do not need to distinguish them. So you do not need to distinguish the type of duct. It's not necessary. Okay, That was tongue development in 11, 12 weeks. Next slide that we will check, we will start checking development of the skin. And next slide will be periderm. Periderm. So next slide that we will check will be periderm. The periderm. I will try to find a good variation of the slide, number 18. So periderm slide. So about the periderm, I will move through the microscope a little bit and then we will talk about the periderm. So we will go through the slide afterwards. I will start with multiple pictures so you can actually understand what we are checking. So periderm slides are as in slides like these. So typically these are slides that show fragments of extremities. Here, for example, you have a developing arm. So arm development here. There are slides where there's also the leg. I need to check, maybe I can see the leg. I will quickly go through the slide variations. Okay, so one type of slide is this one where the arm appears. In the microscope, I will show you a slide where the leg is, where you can also see the periderm. To see the periderm, you need to zoom in on the surface. So you need to zoom to a very, very high magnification. And then you will start to see the periderm. So in these slides, if something is asked about periderm, it will be zoomed in very high magnification. And I will go through the pictures where it's actually zoomed in. Periderm slides are this one. So if you zoom very, very closely to this developing skin, this is how periderm slides look like. So what we will talk about here is we need to talk about two layers of cells. In general, we have first basal layer. So here, this with number 1, this is basal cells. So slide name is periderm. And first, basal cell. So basal cells sit in the basal layer. So it's a basal layer of this developing epithelium. And these cells will be stem cells that will regenerate other cells later. So in basal layer, you have basal cells, as simple as that. Above the basal layer, you see there's periderm cells. So number 2, periderm or peridermal cell. Peridermal cell. So this top layer, the whole top layer is called the periderm. So this second top layer is called the periderm. In your studies, technically there's subgroups of these periderm cells, but for simplicity's sake, if you just call them periderm, it's okay. So in these early stages of development, it's approximately 6-7 weeks of development. Skin is very, very thin. So epidermis has only 2 layers, basal layer and periderm. And some of these peridermal cells are hypertrophic. I will put letter H. That's a hypertrophic peridermal cell. So it looks like a balloon. So it's basically becoming larger. This cell will die by apoptosis. So apoptotic or hypertrophic peridermal cell. I will add letter A. So I will write here HA. That means hypertrophic or apoptotic peridermal cell. So these peridermal cells die off eventually. Some of them will remain, but many of them die off. Other peridermal cells are more flat, so flattened peridermal cells. So flattened or normal, as I would say. So flattened peridermal cells. So these cells are not going through apoptosis. They will stay in the surface of the epithelium and will persist for some time. But if you just label them periderm cells, it will be okay. So it will be an okay answer. So don't mix them up with the basal cells. That's the main point here. Next, what we need to talk about, Below the epidermis, technically, we have the developing dermis. So number 3, developing dermis. In your studies, it's written as subepidermal connective tissue, but you can be a bit more simpler than this, developing dermis. That is also fully valid answer. Developing dermis. Developing dermis. So you can actually write mesenchyme here. That will be also an okay answer because it is mesenchyme. Connective tissue, mesenchymocytes are here. Embryonic connective tissue, mesenchyme. So for the single area, there's multiple names that you can use. And what else is here? Only in this slide picture. It's typically not seen in the microscope. Here I will put X. This X is a lymphatic capillary. Lymphatic capillary. It's only in this slide picture. In most slides, if you get lucky, you could see it. But in most slides, you won't see lymphatic capillaries. They look like normal capillaries with some differences. A much larger lumen, no erythrocytes typically inside, no blood cells. Maybe some lymphocytes appear, but basically no blood cells normally. They look like big empty spaces lined with endothelium. That's a lymphatic capillary. In slides, you could actually see blood vessels. And in this magnification, mainly normal capillaries. In this slide, it's very hard to see a clear capillary. No, in this picture, maybe here, this could be a capillary. Maybe here, I'll put an asterisk, but it's not always visible. Here in this slide, these capillaries don't look that good. There are better slides than this. This slide is good because we can actually see the periderm layers, the basal layer also, and the connective tissue. But with blood vessels, lymphatic capillaries, it's very, very difficult to see the differences. So that's about this picture. Now I will go to the microscope. Let's see something like this in the microscope. So let's try to see this under the microscope. So periderm slide. I will clear this off. And let's check the periderm. So I will go to the microscope and how the slide could be analyzed. So periderm slides are checked in high magnification. First, we always go in low magnification. Basically, this is a developing extremity. Most likely, this looks like it's the developing leg. There's slides from the leg. There's arm slides also. But you zoom in on the very surface where the skin is developing. Let's move here. I will zoom in this area. So I will zoom in this specific area. Yes, there's a question? Number of the slide, you mean? Slide number, on the slide plates, it's number 18. So yeah, so it's labeled on the slide plates with number 18. Okay, let's move on. So these slides, if you want to be very, very specific, periderm slides are taken from human embryos in weeks 7, 8. There's some 6, 7 weeks also, but typically weeks 7, 8. So it's not mandatory for this slide because sometimes this can also be asked in animals, but weeks 7, 8, technically. Technically, weeks 7, 8 is also on these slide plates, but it's not mandatory for this slide. Okay, let's move on. I will zoom in to the highest possible magnification, and let's check the periderm. So, first of all, as you can see, in the highest magnification, everything can look quite blurry, but I will find an area where we could actually see the layers a bit better. So, let's check here. I will go to the zoom so I can actually label some details. So on zoom, what I can do here, so if I use the drawing function, again, we can label basal cell. So we see a basal cell, that's number 1. And we can see periderm, number 2. These peridermal cells are flat cells that sit on the basal cell. So 2 layers of cells. Here it's very, very blurry. So you need to be careful with this. If I show the slide in the microscope in the colloquium, I will try to go to a place where we can actually see these 2 layers. Unfortunately, the artifacts can appear. You can see some of the cells are damaged. So these artifact areas, I won't show you for the colloquium, I will try to find areas where you can actually see these cells. Yeah, about the labels, yes, you can just write periderm. There is no need to write developing periderm. But if you write developing periderm, that won't be a mistake. So you don't need to worry. You can always add developing. It won't be a mistake for embryology. Number 3, mesenchyme or developing dermis. Again, developing dermis, yes? Number 2, developing periderm or peridermal cell. 1 is a basal cell, 2 peridermal cell, 3 developing dermis. So you see mesenchyme there. And inside you see blood vessels. So where I'm writing BV, blood vessels, you see a lot of erythrocytes here, there's blood vessels. There's no lymphatic capillaries. In this area, there's no lymphatic capillaries at all. It's only in a single picture in these studies. That's why it's in your slide descriptions. But mainly you will see normal blood vessels. Mainly normal blood vessels will be in real slides. Yes? Sharing is caring. So yeah, check the cables, but be quiet about this. Yes, look at check the slides. Okay, so that's the idea about the periderm slide. Not that much to check, but what you need to be careful with, I will clear this image. I will zoom out. So in these slides, you can actually see some things that were in the first colloquium. For example, we can actually spot some skeletal muscle tissue. So skeletal muscle tissue can appear a little bit in these slides. Skeletal muscle tissue development. That's a variation for the slide. Also, cartilage, hyaline cartilage appears in these slides because you see the developing limb, developing skeleton, hyaline cartilage also, can appear. So hyaline cartilage, I moved a bit out. So hyaline cartilage is here. So these slides have a lot of other details. So hyaline cartilage, as you can see here, muscle tissue development, blood vessels, this also appears in this slide. But it's just a variation. In these slides, mainly I will try to focus on the periderm associated structures. If this slide is clear, let's move to the next slide. We will talk about development of skin and hair follicles. In your e-studies, the slide is labeled, development of skin and its derivatives, but you can write development of skin and hair follicles. Development of skin and hair follicles. And this slide that I'm going to show you is taken from the rat embryo. So from the rat embryo, it's labeled with number 19 on the slide plates. So developing skin and hair follicles. Slide number 19 on the slide plates. We will see the rat embryo quite a lot in the on the 6th practical class. I will try to find a good location where we actually have the hair follicles. Okay, found it. So this is the rat embryo. I will focus on these areas and I will show you the pictures first, but we will check these areas. This is the lower lip and this is the upper lip. And inside there's big hair follicles. And in your e-studies pictures, that's the same thing. So for rat embryos, you might see this. So you see a upper lip. So this is the whole upper lip. I will change to different color. So you see this upper lip region. So this is the upper lip. And inside the upper lip, you will see hair follicles. So these are developing hair follicles. Developing hair follicles. I will now move to the slide where this is zoomed in higher magnification so we can actually label the smaller structures. So we will zoom in. So I will use this slide and let's go through all the structures that you should know here. First of all, I will label a general structure. This whole surface, that is developing epidermis. So don't worry, I will go through every structure, so don't worry, we'll return to the next slide. So the slide is developing skin and hair follicles. And the first structure here, developing epidermis. So all of these layers together, that's developing epidermis. So developing epidermis. Here, because it's a rat embryo, skin has developed much further. In humans, this takes multiple weeks. Basically, the skin like this in humans looks typically more in the second trimester. In rats, it happens much more quickly. That's why we use rat embryos. This means no weeks. You don't need to know the weeks for this slide. Next layer is here. You see small cuboidal basal cells, so you have the basal layer. So number 2, basal layer. Basal layer with basal cells. So basal layer with basal cells. There's still a basal layer here. And in between, you can see it's no longer 2 layers. You actually have many more layers inside this developing epidermis. These layers in between, we call this stratum intermedium. Stratum intermedium, that's number 2. So layers in between, in the middle part of the epithelium, we call this stratum intermedium. It later will differentiate into different layers of the epidermis, stratum spinosum, granulosum, etc. So all of these different layers will form later. But for now, we call it stratum intermedium. That's number 3. And you can still see there's an outermost layer of flat cells. Flat cells are still here. And you can still label this as periderm. Number 4, periderm. So top layer with flat cells, periderm. So there's still some periderm left. Later it will disappear, but at this moment it's a transitional stage between fully mature skin and this very early development of skin. So this slide shows that another layer has formed, stratum intermedium, that separates the basal layer from the periderm cells. So that's about the epidermis, what could be asked here. I see in the slide descriptions only stratum intermedium is mentioned, but to understand where it is, you still need to know where is the basal layer and the periderm. Second, the connective tissue. Here, this shows, again, developing dermis. So number 5, developing dermis. So number 5, developing dermis. So developing dermis also appears here. Developing dermis. So developing dermis. And if you write mesenchyme, that's also a valid answer. So connective tissue, where skin connective tissue will form developing dermis, developing mesenchyme. Similar to the previous slide. And this whole structure, developing hair follicle. So number 6, developing hair follicle. 6, developing hair follicle. Developing hair follicle. Technically, there's different layers here. Remember from histology, so you had the hair with cortex, medulla, inner, outer epithelial root sheet, connective tissue sheet. These sheets won't be asked in embryology, but still, sometimes you can distinguish them. What else you need to be careful with? In your e-studies, I can see it's written primordia of the mustache in your e-studies. These are so-called mustaches of the rat, or we call these specific hairs vibrissae. So these are specific hairs that are very long, very large. They have big hair follicles, and they are very well developed in rats. In humans, we don't have vibrissae, as you understand. Of course, we can have our mustache or our beard, especially males, but these hair follicles are a bit different. They are much, much larger. These are specific for rats. So if you just write developing hair follicles, that's an okay answer. You don't need to remember the name that's on the list there. So just developing hair follicles, and that's fine. That's what you can see on higher magnification. I will show you now what we can see if we zoom out. So we see there's a slide where you could actually get these original hair follicles. This is the rat's head. In your e-studies description, actually there's more structures than needed there. We will talk about the whole rat's head on the sixth practical class. You will have the whole rat embryo, we'll go through each of these structures. But what you should note, if you zoom in, at the very tip, basically it's the same upper lip with hair follicles. It's the same type of slide. But I will emphasize some of the structures that I mentioned in the list so you can understand this. So first of all, some generic structures. You can understand that number one is the tongue. So that is the tongue logically. So we have the tongue. So we also have the upper lip, lower lip. So I'll write down. So rat embryo head, and it has hair follicle development. Hair follicle development plus skin development. So this is the original slide where we could actually find the skin development and hair follicle development. 1 tongue, 2 upper lip, 3 lower lip. So we see the lips. If we would zoom on the lips, we will again see the same skin development with all 3 layers, hair follicle development. So I won't repeat that. What we can also see here, if it's the upper lip, we have the upper jaw, and inside the upper jaw, we have developing nasal cavity. So number 4, developing nasal cavity. Developing nasal cavity. And for the developing nasal cavity, technically, we can separate the mucosa. So there's the developing nasal mucosa. I will just put the letter M here, developing mucosa. In your e-studies written developing mucosal membrane, but it's the same thing, it's developing mucosa. And also in your e-studies written developing nasal conches, I will put C, developing nasal conches. This is typically not asked in the colloquium. I'm just showing you that in some slides it can appear, but in many slides not actually visible. So, yes? M, mucosal membrane, or M would be also developing nasal mucosa. Developing nasal mucosa, that's M, and C would be developing nasal conches. So developing nasal conche, or as you know, these protrusions of the nasal cavity, developing conche. These 2 things are typically not asked in the colloquium. They are typically not asked. We use another term, I will write down, for nasal cavity development. You can also use a term called, so nose or nasal cartilaginous capsule. This appears in some textbooks. Yes? Do you see some cartilage here? It's a bit blurry. These are the conches, nasal conches. Those are the protrusions of the wall of the nasal cavity. Those are the conches, yes? Yeah, sorry. So nasal cavity development sometimes is called nasal cartilaginous capsule because walls of the nasal cavity are actually made from hyaline cartilage. This term is used interchangeably sometimes in slides. That is just a synonym. If you remember nasal cavity development, that's completely fine. Okay, let's move on. We have a lot of slides to check still. We still have some other details. We also have some bone tissue. Here, we will talk about this also in human embryo slides a bit later, and in the last class when we have the rat embryo, number 5, desmocranium. Desmocranium. What is that? Desmocranium are the flat bones of the skull and the face that develop through intermembranous ossification. When you zoom in, it's very blurry, but you will see bone trabecules. Bone trabecules. I won't emphasize this that much here because we will have the rat embryo and then we will see it in more detail there in the last class. So I won't emphasize this much. There's a lot of other structure to check. But the desmocranium, please remember this term when we talk about flat bones of the arch of the skull in the facial region. So desmocranium. We also have chondrocranium. I will show here. There's a hyaline cartilage plate. Basically, it's the occipital bone. And this, with number 6, this is chondrocranium. Chondrocranium. Basically, it means base of the skull. Base of the skull is made from hyaline cartilage during embryonic development. That's why we call this chondrocranium. Typically, at the lower parts of the skull, you might see hyaline cartilage. Even this fragment is also still part of chondrocranium. Chondrocranium. Chondrocranium is the base of the skull. It develops, it has hyaline cartilage, it will later replaced by bone tissue. And so endochondral ossification will happen there. So chondrocranium, number 6, chondrocranium. Then what else? Some general details. If this is the head, then you can logically understand that we have the brain development. The brain develops there in the skull cavity, brain development. About brain development, we will have a separate slide next week, brain development. So I won't go into the details what's there, just brain development is fine here. So some structures are mixing. We don't really see tooth development that well. We don't see that many glands here, but we will see them better in the last 6th class. So in this slide, we cannot really see the glands clearly anywhere, not in the oral cavity, not in the nose, nowhere. So it's a bit, it's a problem with the magnification, also a bit of a problem to the resolution of the picture. But some structures, as I mentioned, are not always visible, and these are typically not emphasized in the colloquium. Who can answer me what is here with number 8? What is here? What do you think? Yes? Developing vertebral column. Yep, correct. Developing vertebral column. So here you might even see part of the neck and you might see developing vertebral column fragments. Vertebral column. Okay, that's it. So that's a brief overview of this type of slide. So it's a very specific slide. I will briefly only show the skin development and hair follicles in the rat's head because we will see the rat's head in the last class. So don't worry about it. We will return to this slide during next classes. But okay, in the microscope now. So I've zoomed in here on the rat's head, specifically where the hair follicles are developing in these areas. There's the lower lip and the upper lip here. I will zoom in and let's see the structures in the microscope a little bit. I will zoom into one of the areas, not like here, for example. Then I will briefly talk about the details. So what's interesting about this specific rat's head? So it is even more developed than the slides that we had in the Eastides. So if we zoom in, we see something like this. And what we can emphasize, again, we have developing epidermis. I will use maybe red color that stands out. So developing epidermis, that's number 1. So developing epidermis, and inside this epidermis, again, you have the basal layer. That's number 2. Technically, there's even more differentiated layers, but for simplicity's sake, let's take the middle layer, stratum intermedium, that's number 3, and layer of flattened cells, periderm, that's number 4. Same labels that I used for the picture. So 1, developing epidermis, 2, basal cell or basal layer, 3, stratum intermedium, 4, periderm. Then, if we go into the opposite surface, we have connective tissue. That's number 5, developing dermis. or mesenchyme. Developing dermis or mesenchyme. So developing dermis or mesenchyme. And inside, we see developing hair follicles. That's number 6, developing hair follicles. You can actually see fragments of the hair, these epithelial root sheets, and even a gland. This is a sebaceous gland. But this won't be asked in the colloquium, so histological structures of the developing hair won't be asked. You just need to remember hair primordia or developing hair follicle. So hair follicle primordia or developing hair follicle. That is the term you should remember. Yes? Number 5, it's here, connective tissue. Here, number 5, that is developing dermis. I will point to also other areas. That's number 5, developing dermis, where the connective tissue is, developing dermis. Okay, I hope you have the idea. We have 2 more slides to check. They're quite, 3 slides. Let's be quite quick because we don't have much time left. Let's check next slide, nail development. Nail development. I will also put it on this nail development. Let's start with this slide. I will go through some variations. So nail development. Nail development, that's the next slide. That has number 20 on the slide plate. So number 20 on the slide plates. Okay. Okay. That's the microscope. Okay, let's go to the picture. So if we check the picture in these studies, you should check only this single finger. It's the thumb. So this finger, you need only on a single finger, you might see a nail or developing nail. And why? It's because of the section. So many cases you have a hand in frontal section. So you see hand with sometimes 5 fingers or 5 finger fragments. If you cut the hand in a frontal section, look which finger is positioned in this way where you can actually see the nail. That's the thumb. And that's the reason why you only see some parts of nail development on the thumb. So only this finger. So I will move to the slides where we can actually see the thumb in higher magnification, like this one. It's the thumb. What are the structures here? It's very similar to histology. First of all, you have the nail plate. Number 1, nail plate. So slide is nail development. And first nail plate. Number 1 is the nail plate. What you need to be careful, nail plate can be missing. So it can be missing. If I move to the previous slide, like this one, there's actually no nail plate. It has fallen off. Sometimes it can be lost due to artifacts. But nail plate is fragmented and typically eosinophilic, bright red, bright pink in most slides. If it's there, it can be missing, so be careful with this. Below the nail plate, you have the nail bed. That's number 2. You can always add developing, so nail bed. So nail bed, that's the epithelium that sits below the nail plate. Number 3. So here you see that the nail plate goes into the underlying tissue. That is the nail root. So number 3, nail root. So you can see the nail root very well. And the epithelium from which the nail grows, we call this nail matrix. Nail matrix. So there's nail matrix also here, nail matrix. The problem is it's very hard to see the nail root. So it's very, very small, but you can see the nail matrix. This darkened epithelium, that's the nail matrix. And the nail matrix sits all around the nail root. This epithelium all around, it's the nail matrix, all around. All around, that's the nail matrix. Not only at the tip, but all around. So nail matrix. In your e-studies, nail plate is mentioned as nail lamina, but it's the same thing. Nail plate, nail lamina is the same thing. It's bright red because it's keratinized. What else can we see here? Don't forget, you have number 5 developing epidermis and number 6 developing dermis. You still have some skin around. So 6 developing epidermis and sorry, I will clear this. Mistake, delete, sorry. Number 5 developing epidermis, yeah. And number 6 developing dermis. So don't forget the skin is still here. You see there's question in the slide name only nail, or should we also know their weeks? So the slide, you can use just nail development because there's some variable weeks. If I check here, in here, the slides are given that they are 11, 12 weeks old. So in human embryos, 11, 12 weeks. Mainly it's taken from human embryo in 11, 12 weeks. But if you forget for this slide, that won't be a big mistake because there's some variations also. Okay, what else? Who can tell me what's happening here? Where I put the red circle, what is there? Yes? Ossification center or endochondral ossification? Yes, so ossification centers can appear. So here I will just put number 7, ossification center. It can appear in some slides, ossification center. What do you think? What is here? Yes? In that opening here, where I'm pointing at, what is this hole? It's not condensed mesenchyme, it's something else. No. So think logically. So it's a finger. Distal phalanx, proximal phalanx, what's in between? Joint. So joint cavity, developing joint. So you have the joint here, joint cavity. No, it's not intervertebral space, so developing joint cavity. So yeah, thank you for the guesses. But here, think logically. So if you're actually seeing fingers, don't forget anatomy, phalanx, they're connected with joints. So joints are here. So that's the idea here. So these are just some specific details. Main points of interest are in this part of the slide. Main points of interest are here. Most slides are similar. I won't repeat all the slide variations. I will go directly to the microscope because we don't have much time left. So I will clear the drawings and let's check the microscope. So here, this image again shows nail development. And it's, again, a thumb. You might see other fingers, but you should try to find just the thumb. So one of the fingers should appear. So there's a lot of other structures, but we mainly focus on this area. If you zoom in, again, we can check nail plate is reddish color. Here you see fragments of the nail plate. You see the nail bed very well. So the nail bed is this thick and dark epithelium here. You also can see the nail root and the nail matrix. Nail root is this plate on top that goes into the nail, deeper into the tissue. And so nail root goes here, and then nail matrix is this epithelium around the nail root. So that appears again in this slide. Of course, we have some mesenchyme. On opposite side, you might see epidermis, dermis development. And there is also some fragments. So some hyaline cartilage appears here, joint cavity, etc. So just a variation of the slide. Other variations you will need to check on your own. Let's move on. Yes? Here, this red fragment. The nail plate is damaged here. It's very, very small, very limited. Nail lamina is the same as nail plate. Yeah. Okay, let's move on. Next slide that we need to check will be development of the lower part of the face. I will be very brief because we still have to check the adrenal gland slide, but development of the lower part of the face. That slide with number 21. So this slide has something from everywhere. So let's use this slide. Yeah, okay. So it's a bit damaged, but I will use the picture mainly. This slide is very similar to the picture in your studies. So development lower part of the face is described in, oh, find the slide, this slide. Basically, what is the slide? Remember, we had tongue 6-7 weeks. You had also the lower lip, Meckel's cartilage. So imagine you have the tongue in 11-12 weeks, and you have also the lower part of the jaw, which is much more developed. So it will be very similar with some differences to that 6-7 weeks old tongue slide, the lower part of the face. But let's talk. Again, their surfaces. First, oral cavity epithelium, that's number 1. Oral cavity epithelium, that's on the surface. I will write it in the chat. So oral cavity epithelium. And below, mesenchyme or ectomesenchyme, because it's the face, so mesenchyme or ectomesenchyme. So mesenchyme or ectomesenchyme. Who has an idea what's happening here? Yes? Which type of tooth development is this? Yes, early tooth development. And if you want the stage, this is the Bell stage. So early tooth development and Bell stage. I won't ask you the different epithelia here or the tooth, dental follicle, dental sac, dental papille, all the 4 epithelia. I won't ask this, but if you remember early tooth development, that will be okay. Next, you can understand this is the lip. So here, this number 4, this is the lower lip. So lower lip. Unfortunately, this lip has been damaged. So there's damage to the tissue. You don't see the tip of the lip, but you can see that on one side, again, number 5, developing epidermis. So that's number 5, again, on one surface. Connective tissue here, number 6, developing dermis. And number 7, developing hair follicles or hair follicle primordia. Hair follicle primordia. Hair follicle primordia. So again, 5 developing epidermis, 6 developing dermis, 7 hair follicle primordia. Inside, you might see reddish developing skeletal muscle tissue. So these red dots inside, developing skeletal muscle tissue. So number 8, developing skeletal striated muscle tissue. So developing skeletal striated muscle tissue. What else is here? Inside, you can actually see nerve fiber bundles. Some nerve fiber bundles where I'm putting the labels here. Number 9, nerve fiber bundles. So there's some nerve fiber bundles visible here. Not in every slide, but in some, yes? I can zoom in. You can see nerve fiber bundles. They look like this. That's number 9. Then, what else we can see? We can nicely see intramembranous ossification. Number 10, intramembranous ossification. So intramembranous ossification is here. Number 10, intramembranous ossification. Inside the bone tissue, I can zoom in. Please repeat intramembranous ossification a little bit. about bone cells, osteocytes, osteoblasts, even osteoclasts. Bone cells, repeat that from histology. Osteoblasts on the surface, inside bone tissue, osteocytes, as you can see, and osteoclasts like this one. This is a nice osteoclast here. A lot of nuclei, pinkish cytoplasm. But that you need to zoom in very high magnification. If it's zoomed out, you won't see it. But remember from histology, please, bone cells. What else? You might see some glands. For example, you can notice here with number 11, there's some glands inside the lip. These again are developing minor salivary glands. Developing minor salivary glands, also called labial glands. Or glandula labialis in Latin. So if you zoom in, you might see some glands inside the lip. These are developing labial glands. So developing minor salivary glands, labial glands. So minor salivary glands. And in general, that's everything. As I mentioned, I won't ask you specific tooth structures like the lamina dentis, inner outer enamel epithelium, then stratum intermedium, stellate reticulum, dental papille, dental sac. These structures won't be asked. Yes? We see them, but I won't ask them because you already have hundreds of structures to memorize. In the tongue, you mean? Sorry, I misunderstood you. So we don't see the tongue. Tongue should be here. The tongue should be there. No tongue is here within the slide. It's just cut off from the surface. No tongue. That's it about this slide. I will be super brief in the microscope because we still have the adrenal gland and I don't want you to stay after the class. I will very quickly show the microscope. I want to emphasize that many structures, but basically, again, so the slide is big. But here on one side, you might see bone tissue. So there's intermembranous ossification here. Bone tissue appears in the slide. So bone tissue appears in the slide. There is even some remnants of cartilage tissue. Some Meckel's cartilage can appear, but normally it's not there. It's a rare slide variation. It's very, very rare. But Meckel's cartilage is not typically seen in these slides. Then early tooth development. Here the tooth is not that well looking, but still this is early tooth development. We see oral cavity epithelium on one side, and we see the lip. We can actually see the lip a bit better. On one side, you have the oral cavity epithelium. On the other side, the epidermis development. Inside skeletal muscle tissue, as we can see, on skin side, we have hair follicle development. On lip side, minor salivary glands or labial glands. We can see the lip structure here a bit better. If we move out, of course, some nerve fiber bundles can appear. You need to zoom in high magnification. Again, skeletal muscle tissue, bone tissue, etc. So it's just a variation of a slide that combines everything together. So, colleague, where are you going? The class ends after 10 minutes. Then you can leave. So if you have a reason, of course, you can tell before the class. So it's not really correct to do that. Okay, final slide, development of adrenal gland and metanephros, weeks 11, 12. So development of adrenal gland and metanephros, weeks 11, 12. That's the final slide. Let's carefully go through it. So again, I will start with the pictures. So slide name, development of adrenal gland, the metanephros, weeks 11, 12. So development of adrenal gland, the metanephros, weeks 11, 12. So you have different slide pictures. And this slide is a bit similar to the ones we saw for the first colloquium, but a lot of differences because it's a much older adrenal gland and metanephros, development of adrenal gland. Weeks 11, 12. So what you should distinguish, let's start with the big parts. First, adrenal gland, so developing adrenal gland. Second, developing metanephros, or just metanephros. So one, adrenal gland development. Two, metanephros. So far, so good. Around each of these organs, you might see developing connective tissue capsule. So you might see mesenchyme around that's developing connective tissue capsule. So developing connective tissue capsule. Tissue capsule. So it's around both of these glands. If you write mesenchyme, that's also an okay answer. You can write mesenchyme because it is mesenchyme. Next, I will clear these drawings. Let's zoom in. About the adrenal gland. You see adrenal gland has two distinct parts, but it's still made only from cortex. Inner part of the adrenal gland, that's number 4, fetal cortex. So fetal cortex, that's number 4. So 4, fetal, or also called primary cortex. And number 5, it's mature or adult cortex, or sometimes called secondary cortex. So primary cortex is in the middle, secondary cortex on the periphery. So you have primary cortex, secondary cortex. No medulla yet, no medulla, remember this, no medulla. I will write in capital letters, no medulla. Remember this carefully. Inside primary cortex, outside secondary cortex, no medulla. Then what else? If you check the adrenal glands, I will show another slide variation. If you check inside the adrenal glands, you will spot sometimes dark cell groups that move deeper inside the adrenal gland. These are migrating neural crest cells. Migrating neural crest cells. I'm checking your slide. It's not given there, but these are neural crest cells. This is the early start of medulla. It's not in all slides. You can remember this. It's not in all slides, but migrating neural crest cells. Here, where I'm pointing, number 6, it's inside the adrenal gland. So these dark spots, that's a migrating neural crest cell group inside the adrenal gland. From these cells, the medulla will develop. So medulla will develop here. Number 6, migrating neural crest cells. This is a special type of structure. It's not always visible, but sometimes for those who might get the exam, this might be asked. That's why I'm telling you this. Yes, yes. It's just here you can see them better than in the previous slide. That's why I'm showing you this. Okay, that's everything about the adrenal gland. Next, I will go to the previous slide, this one, about metanephros. You can distinguish different parts. First of all, you should distinguish that there's a big opening. So number 7, this is the renal pelvis. 7, renal pelvis. Renal pelvis, this is the inner portion of the kidney, the pelvis of the kidney, renal pelvis is here. And actually, if you see this renal pelvis, then what we can see here? We can see a lot of mesenchyme. Number 8, where the medulla will eventually form. Here you have mesenchyme. So there's a lot of mesenchyme present. Mesenchyme, quite a lot of mesenchymes here. And here, what's happening? This outer area, you can label it in multiple ways. I will write all the different ways that you can use. First, you can write developing cortex, or developing kidney cortex. That's one way to write. Second name, developing nephrons, because there's nephrons that are developing here. And third way, nephrogenic blastema. So nephrogenic blastema. All names mean the same thing. Basically, it's the outer part of the kidney that's developing, and first nephrons are forming here. Nephron formations happening here. So this slide number 6 is not that well visible. I can try to show it, but it's a bit problematic. Like here, it's a small dark dot of small cells. You can only see it very high magnification, unfortunately. Not in this slide. But 9, so that's basically developing kidney cortex, or this differentiation of nephrons, so developing nephrons. There's different stages how the nephrons develop, but you will read this on your own theoretically. What else is left? We need to finish the class soon. Sometimes near the entry point of the kidney or the metanephros, you might see dark cell groups. And this number 10 is called primary lymph nodes, or primary lymph nodes. So these are lymphoid cell groups that can be found next to the metanephros, sometimes also close to adrenal gland, but typically closer to metanephros. So primary lymph node slides. In some slides, so I hope you got this. In some slides, these primary lymph nodes can look like this, so big clumps of tissue around. So big clumps of tissue can appear in the slide. Also, be careful structures like the vertebral tissue, but we will talk about the liver next week. Very quickly, I will show you the mic. Again, problem, very large slide. So we see the developing adrenal gland and the metanephros here. Adrenal gland has fetal cortex on the inside, secondary cortex or cortex on the outside. While if we check the metanephros renal, if we zoom inside the adrenal gland, you will see small neural crest cells. Only in high magnification, low magnification, you won't see them. It's also sometimes rare, it's not in every slide. Migrating neural crest cells. And the final thing, primary lymph nodes. Here in this side, we don't see them that well. If we move to the opposite side, I hope, we see another one. There's some fragments that could actually be primary lymph nodes, some fragments near the vertebral column, but here it's not really that well visible. So here the bonus is you might see the vertebral column in some slides. Vertebral column can appear. Okay, thank you, colleagues. This has been a very long class. See you next week. So if there's any additional questions, we can discuss them in the consultation times. Okay, those who have, I hope everyone that's here has written their names under this paper. So those who are on site, please write your name so we can check your attendance. Yes, I will tell you the result. Those colleagues that are online, thank you very much. The class unfortunately is over. We can discuss this a bit later when we have the consultation time. So sorry, colleagues, we need to finish. Okay. Thank you very much and goodbye. So see you later. Consultations are on Thursdays. So on Thursdays, we have the consultation times. On Thursday afternoons. so we can discuss this. You can write an email. We can also check separate consultation times if needed, but write an email. We can discuss some of these slides in more detail. So thank you very much and goodbye, colleagues.